Okay. Uh, so hello everyone. I'm Min Hao. Today I'm going to share a paper titled Universal Cell Embeddings, a Foundation Model for Cell Biology. And let's first learn something about the author information. The first authors of this paper is Yanei and Yusuf. They are from Stanford, Department of Computer Science and Biomedical Data Science. The corresponding author of this paper is Jur Leskovec. He is very famous because he designed the algorithm of node-to-vector and has over maybe 150,000 citations. And he has done a lot of graph-related work, and is a very famous scientist. And this paper is published on the end of November last year and is currently on bioRxiv. According to its structure, I think maybe it is submitted to maybe Nature Biotechnology. I'm not sure, but I think it's a subjournal of Nature. Yes. And before we dive into the details, let's first learn some background of the single-cell foundation models. In short, it can be divided into two parts. The first type is the pre-training, which means we have the single-cell transcriptome, and we use that to train a single-cell foundation model, and we can get an embedding of the genes for each cell, and then we design downstream tasks according to this embedding. And there are a lot of work for pre-training, and all of those are pre-trained from scratch. And another type called prompt learning. It means we design some prompt, like we type in some words in the GPT-4, and based on those previous foundation models, and we use some prompt to make it adapt to our specific task. And UCE belongs to the pre-training type. And here is an overview of two types of single-cell foundation model. The upper picture is the pre-training part, and the lower is the prompt learning part. And for the pre-training part, we can see that we have the single-cell RNA sequence data. Hello, okay. And after passing through a kind of block, encoder block, we'll get the embedding E here. And we may design different tasks. We have two kinds of tasks. Do you see the screen? Okay, let's move on. We have two types of tasks. One type is the generative task. We may, for example, we may do some cell generation, and we also have this kind of task. This embedding is passing through some fine-tuning block, and we may do like cell type annotation. We may have the drug sensitive prediction, and we also have some like this, the in silico perturbation, and this is the gene network prediction, and there are various downstream tasks, and all of those tasks are designed according to this embedding. This is the procedure of the pre-training type. And this is the prompt learning part. We may see that we have a pre-trained foundation model here, like GPT-4, like Llama, like others. And we have the single-cell transcriptome, and we previously designed some prompt, like, could you please generate the information of some genes for me? And that gene is dynamic. We need to change it according to our input. And then it will give us an embedding. We use the single-cell transcriptome combined with this embedding to get the final embedding. and we add some task-specific adapters, such as MLP, logistic regression. We use that to do different downstream tasks. So this, for this kind of prompt learning task, we don't need to pre-train or fine-tune. We just design some prompt to make this foundation model adapt to our specific task. Okay. Since for the single cell foundation model, currently there is only one work, just like this picture. The input is first is the cell by gene matrix, and it will use GPT 3.5. They use GPT 3.5, and they first design some fixed prompt, just like what we type in the GPT 3.5. And actually, they will generate a response, a text, right? And they use this text. They use the text to extract the embedding. From origin, you will have. Because it will generate a lot of information based on this foundation model, right? Based on GPT. And they use this output, they use this response to generate embedding. Actually, if we use the GPT API, it will give us not the words. It's another kind of embedding. Yes, so we just need we just keep this embedding aligned, the dimension aligned with our cell by gene matrix.
So, why we need to like fit the gene name to the gene? What's the goal of like adding such from embedding to the cell by gene?
For this paper, they say that a very simple this is a very simple work, but it can achieve a rather satisfying result. So you mean why they want to add this one?
What do they want to extract and convert?
Actually, I'm not quite sure. Yeah, I just see this work, and I think this is also interesting, so I listed here. But for the details, maybe we can talk later.
What's this
here? I think this. Sorry, I'm not sure. Yeah, sorry. Okay, sorry.
They use the foundation model to generate features.
Is there a subset of the original?
I think it's not the subset of this matrix. Maybe. Yeah, it's like the foundation model can both give the feature embedding and cell embedding, because it has two arrows here. Yeah, but here they use the generative language model like GPT-4, like Llama, but they don't use the pre-trained single cell foundation models.
So the embedding is not related with the cell multiple features matrix. It's just a general prior knowledge.
Yes, from the foundation model. Yeah.
Maybe two questions. So maybe on the previous slide, what do you mean for the foundation? Do you mean the both pre-trained model and also the prompt learning model?
Yes, foundation models are first they need to be pre-trained on large data set. And then they have the fine-tuning part. Yes, and both the pre-training and prompt learning are foundation models, but just here they use existing foundation models instead of they pre-train a new model.
So for the pre-trained part, they use the previous other pre-trained model.
No, they train from scratch. They just borrow other model structure, but the data collection and the pre-training are from scratch.
So you mean the prompt learning model also pre-trained?
No. Prompt learning just use previous foundation models like GPT-4, like those large language models.
They do not.
They do not
the
overlap.
data set.
Yeah, they don't need to pre-train the model.
Then what do you mean for the prompt?
Prompt is what you input in the language model. Like you type some words, that is called prompt.
Yeah, I think maybe a little different from the GPT generated prompt or as the input. Is it right? For example, if I put some glioma specific genes and ask, whether it is in glioma stages, which is the paper-based knowledge, would this prompt learning model understand this kind of sentences?
This prompt learning part mainly borrows the previous knowledge from the generative pre-trained language model. So
if the generative pre-trained model do not have this knowledge, they may not understand.
yes, they for this part, the key idea is to design different prompt to extract information from the large model. You just need to design different prompt.
Design different prompt.
Yes, maybe ask different questions, to be short. Yeah.
This is what we need to do to prepare for the data set for the prompt learning models.
From the data set.
Yeah. I mean for the prompt learning model, if you input some knowledge specific sentences, the model may not understand unless you use a GPT-4, some internet based.
No, I think we can just want to explain this. What is the pre-training? The pre-training is trained from scratch. Use the model structure borrowed from GPT-4, GPT-3.5, and then use the data collected by ourselves or by the researchers to train from scratch. And for the prompt learning, it's just like, do you know the POE? POE, I think all of us have used the POE. The POE has also additional item about the boat. You can generate a lot of types of boat for the English teacher, for the lawyer. And this is the prompt learning. The prompt learning is just like distilling some knowledge from the pre-trained model and to accurate in this part as an expert in some specific area. This is prompt learning. And in here, I think Minhao just try to want to explain about we have trained is foundation model use all kinds of cells from, for example, the mouse, the human, and the monkey. And then we just want to focus on the diffuse, adult diffuse glioma. And we should use the prompt learning to input some keywords like for example, the single cell, what is the single cell like in glioma? And give it to the foundation model, and the foundation model will more focus on the generative about the glioma related area or some content. This is the prompt learning.
So what's the input and the output? Any method?
You can do the prompt learning as a fine-tune, like fine-tune process.
Yeah, it's kind of mixed with fine-tune methods. Yeah.
The kind of mix method you, for
So
example,
we need to input some information.
no, we just give some prompts.
Yes.
Because it has already pre-trained on the glioma data.
This
We
one
just-
we have the pre-trained on the glioma data.
the foundation model to
Oh,
the equivalent.
I see.
Yeah.
So,
We just want to split it from the whole foundation model.
so it is like the second step for the glioma-based pre-trained model?
Yes.
I see, I see.
Yeah, I think maybe I can ask one question. So as you mentioned here, they have many pre-trained model like SC-BERT. I think we have learned it before. Also G-Informer and also SC-GPT, SC Foundation, so many. Then why we still need another pre-training model, UCE?
Yes, I think this is very good question. We have various and a lot of single cell foundation models. Maybe from 2 years ago, from 2022, people start to use this kind of maybe the BERT or transformer or those model structure to train the single cell foundation model. Till now, the trend is that previously, for example, SC-BERT is the first model. It just used 1 million cells. But for example, UCE or SCGPT, they use over 30 million cells. So the data set is maybe 30 times larger than previous. This is the data. And besides, they also add some like tricks, or they add some methods to deal with the input so that it can first adapt to various tasks and perform better. And then besides, a recent trend is that they want to utilize other omics like proteomics. They try to unify those omics information together to help the single cell foundation model get better result. And this UCE just to use a protein language model, combined protein language model together to train a single cell foundation model.
Okay, yeah, then you can show us the details, I think.
Yes, yes. And then since we have the embedding, a very important thing to notice is that for one gene in different cells, the embedding we get is different, which means maybe for one gene in different cells, their information is different. I think that's just suitable to our prior knowledge, right? And so current single cell foundation model has several issues. The first is that the gene length issue, which means, for example, I have a data set. Maybe this data set, we have 20K genes, but I have another data set, maybe it is 25K genes. However, previous foundation model can deal with fixed length of genes. For example, SC-BERT, they can deal with 16K fixed genes. You need to do a match. Just like look up the dictionary, if you have these genes, you keep it up. If you don't have that, you need to put 0 in it. And for example, as a G-Informer, the length of it is around 1000, and it will cut the first 1000 expressed genes to used to train the model. So the gene length is we need to pre-process it before we feed into the foundation model. So this is the first issue. And the second is the species issue. Since although we are working on human genes, but they are still like mouse, like chicken, like zebrafish, a lot of different species. And the previous foundation model can only handle human genes because of their method of the gene names, because they need to treat gene names as some information, and the gene names are fixed. But however, for example, mouse, the gene names are different, so they can't handle those species. And finally is the fine-tune issue. After they pre-train the model, they give the weight to us, and if we want to use it on our own data set, we need to fine-tune it, which means we need a lot of GPU to first extract a subset of our data set to fine-tune it and then use it on our own task. So those three issues make previous foundation models, I think it's a little bit hard to use. So those three issues are all UCE contributions. They try to fix the gene length issue, and they can, I may move to the next slides. They give us a foundation model called UCE. They can generate the embedding of all species without fine-tuning. I think that this is the most important contribution for this paper. Okay.
So what do they mean by all species? So let's say maybe tomorrow I find a new species.
No, sorry. This all species means almost all species. Okay, almost all species. Yes, actually, it provides around maybe around 10 species is provided. And if you have new species, you can create the embedding according to the tutorial. Yes, so they claim that they can deal with all species, but I think for most of the species, they can handle that. Yeah, because we have they have a website, and you can download the maybe the protein information from the ensemble, that website. And another contribution is that they provide a data set called IMA, and it is very simple. It's just created by applying the UCE. We after pre-training that, we apply it on 36 million cells, and we can get 36 million cells of embedding. And they claim that that is another contribution. Yeah, it is. It has a lot of new cell types and a lot of tissues and 8 species. Yeah, and okay.
Normal cells from different degrees.
I think most of the cells are normal cells. Yes, normal cells. And here is the overview of their model. And compared to other foundation models, it has several changes. The first is the embedding, and another is the training scheme. And let's first see the input of it. The input has 2 parts. The first is the expression part. First, we have a lot of, this is one cell, this is one cell. We have a lot of genes, and some of them are 0 expressed. Some of them are non-zero expressed. For those non-zero expressed genes, they may rank them and do a weighted sample with replacement. So we may like here, we may sample gene 2 for several times, right? We may sample gene 2 for several times. And those sampled gene expression values are grouped by the chromosome. So the gene expression value that belongs, the gene that belongs to the same chromosome, their expression value are grouped together. And they also do a sort, they also sort them by the genome location. And then this deals with the expression value. And another important thing is that how to utilize their gene names as information. Like SC-BERT, they may use, previously I said gene to vector. Maybe it's difficult to understand. But here, they use the large protein language model. So for each gene, they transform it to another kind of embedding. Just simply use this language model. And for the details of these 2 parts, I will mention it in the next slides. And let's first see the final input. We have sampled several genes together with their protein embedding. So it's G2P2, G2P2, and G3P3, G1P1. We have some brackets here. The brackets represent one chromosome because we've grouped them by the chromosome. And then we add a special token here. Actually, it's just a randomly initialized token. But after passing through all of the model, it can capture the whole information due to the attention mechanism. And this is also popular used in the BERT pre-training scheme. No, I don't know. And it's hard to say. Yeah, they just introduce their method and see the result. I think for those kind of not yet. Not yet. Yeah, they just first give a kind of method. Yes.
They didn't mention any reason why they
No.
no.
Yeah. No.
I have a question. You just mentioned that you use the protein embedding to reshape the or reconstruct the vector, the token. And
Yes.
how do you rank the tokens? I mean, when you use the bootstrap to extract some genes and you group each gene in one chromosome in, but the chromosome do not have the sequence or rankings.
Yeah. So they sort them by the genome location. Yes, we can see.
Chromosome genome.
No. It's different. We may see this slides. They use a protein language model designed by Facebook called ESM2, and the input of this model is the protein sequence, and the output is protein embedding. So there is one thing that we need to convert those gene names into protein sequence. So there is a website called Ensemble, and it provides a lot of species the profile in a FASTA file. And I just give an example. It's like this. Here is the names. It's chicken, right? And it has two information. The first is MT. It represents the mtDNA chromosome. It's a chromosome of chicken. And the next step is the 2824. Here, I use laser. Here, the 2824, it represents the genome location. Actually, for the biological knowledge, I'm not quite sure, but these two information provides the chromosome and the corresponding location of one gene. And then for the gene, it can convert it into this protein sequence. So it's just a very simple mapping from the gene to its protein sequence. And we can also get the chromosome and the gene position of that gene. So we can use the result to get this final input.
This is interesting, but for the genes, they are for most of the genes actually have different isoforms, which means they have actually different protein sequences. So it's not a one-to-one mapping, but one-to-many. So how do they handle this?
I think maybe this is I think they regard this file or this website as the true label. So for each gene, maybe they just convert it into. Oh, you mean those are different?
Yeah, they are
protein sequence?
Yeah, more than one.
Oh, sorry, I don't know this. I may check their code later, but I think in their style, maybe they just choose the first one. I will update it later, maybe. Yeah. I don't realize that they are different protein sequences. Yes. And anyway, they get the final input of one cell. This is the final input of one cell, and it's just like the blue box here. And for the training scheme, for the pre-training scheme, we may go back to here. We get the input and fed into the model.
Yeah,
Okay.
here just the chromosome A and chromosome B. What's the sorting of the
Oh,
ranking?
you mean the rank of the chromosome,
Yeah.
right? Oh, I think they don't give the explanation of the rank of the chromosome. Yeah, I think it's just inside the chromosome. They have a
Yeah, inside.
yeah, they have a sorted
Yeah, the outside.
yeah, but outside yeah, okay, I may.
Does the outside have the position embedding?
No, they don't have the embedding for different chromosomes.
Two questions.
Okay.
Yeah, first of all, what's the meaning of the weighted sample?
Weighted sample means that
The sampling by the weight of.
yeah, if the expression value is large, and then they will give a higher weight to be sampled.
So do they do the down sampling or the up sampling of parameter?
I think it's obvious down sampling. For here maybe we input maybe 20K genes, and after the sampling, here we only have 1024 genes.
It becomes smaller.
Yeah, it becomes smaller.
Yes.
But I think
to the spread. Not down or up sampling. You can say 2 G1 G1 from the chromosome A.
yeah, but here we have maybe 20K genes. Here we only have 1000 genes. It's duplicated.
Somewhere. Maybe the value is.
No, for those zero expressed genes, we don't sample them.
Yes. So how decide how do you decide the up sampling or down sampling, because with this spreading value maybe it's small.
Yes. Yeah. Okay, I get your point.
The spread to sample the data from. See.
So what about the meaning of the chromosome? Because you mentioned that there are many species. Are they the chromosome from different species or just the A and B, the abstract or something?
Oh, here A and B represent different chromosomes. If you have, for example, one pre-training, but for one cell, the species is fixed. This represents one cell.
One cell.
Yeah.
They train for all the cells with
Yes.
different species.
Yes.
Then the chromosome should have different species or
They just
species?
they just group the genes belongs to the same chromosome together. Yeah, but they don't add chromosome information. They just group them together. Yes,
Species.
by chromosome.
For example, if you input the human cell, then there exists the chicken chromosome, or just only the human chromosome only here, or it is just the human chromosome when you train the human cell.
They don't add chromosome information here.
I think I don't know. This is the biological knowledge. For one gene in human and chicken, that the gene For gene A and gene B, in human, maybe they are in one chromosome, but in chicken, they in the same chromosome. I don't know.
This belongs to we should first do a mapping to get the chromosome information and the position information here. We should first get the result, and then if for one cell, if the genes belong to the same chromosome, we group them together. Is that okay?
Oh, so you mean that the chromosome also have the species information?
Chromosome also have the species information.
I think what Minhao means is that given the cells, the species already determined, then he can determine like which species it is and which chromosomes the genes are.
Yes,
I
because
think.
be-
But
yeah,
then, yeah,
because I don't understand your point.
the different
Okay,
species
yeah,
are
let
not
me,
comparable basically. Chromosome, for example, for two genes, okay.
Okay.
The two genes maybe in human, maybe two genes are on the same chromosome and they are neighbors. But in the chicken, maybe they are still on the same chromosome and still neighbors. Maybe they are not. Maybe they are in different chromosomes. But there's also situations where actually one of the genes not exist in the chicken at all. So there are many situations, I think.
Okay, I know. So so they first extract genes from the cell, and based on those extracted genes, they may look up the file. They may look up this file to get their position. So if, for example, if we have two genes, and in human and in chicken, maybe in human they are in the same chromosome. So here, if this is human, and it will of course belong to the same chromosome. And in chicken, I think the result is they will belong to different chromosome. Yeah, I think this is just this file will already provides us with the information. Yeah.
Species.
Yes.
So like human chromosome 1 and chicken chromosome 1.
Yes, and not correspond with each other,
Would,
actually.
would they be treated the same?
Could you paraphrase your question? Sorry.
So if one cell is human cell and the other cell is chicken cell, and from this file you know that the grouping then
Yeah.
produce
For this file, we can know the human grouping and chicken grouping.
So the first group of the human cell, is it well aligned with the first group of the chicken cell?
Is it well aligned?
Or do you just assume that it is
I think I'm still confused about your question. Because yeah, they have during pre-training they have different species, and your question is if I think in my understanding, if they have for example, if chromosome A, if this group, we don't say chromosome A or chromosome B, if this group is belongs to human, then we can have the we can group them. Yes.
So this model.
No, sorry. I get your point. For different cells, this is different because this just give us illustration of one cell.
Species.
No, because they don't add chromosome information. They just group them together. They just group them together.
So
Like, does not matter. Yeah, like we have this is the first group and this is the second group. And for those separate, we just add a separate token here. We don't add any chromosome information here. Using this kind of file. The gene names and the corresponding species, and they find the chromosome information. This MT is the chromosome, MT chromosome, and this is the gene location.
No matter how can just the protein embedding from the ESM2.
Yes, after that, we can convert the gene names into protein embedding, and we combine. This G2 is the expression embedding, and this P2 is the protein embedding. We combine them together.
Concate.
Yeah. Concatenate. Here, it will generate the embedding of this cell. Right? Because all of the foundation model will give us an embedding. Right? We have the expression part and the protein embedding part, and we concatenate them together, and we can get the embedding of this cell. That is the next part. The pre-training scheme. Given previous input, just what we said before, we can get the embedding of this cell, right? And they also, from this cell, for the pre-training part, they will mask 20% of the expressed genes. Mask means sample them and then change the expression value to 0. Sorry, it is a mask. It is a mask, but it can be regarded as 0. Like here, G1 and G3 are expressed genes, but here we mask them. And then we sample a lot of 0 expressed genes. Like here, G7 is 0 expressed genes. And we just use those protein embedding. We don't use the expression value because we've already masked them. We just use the protein embedding combined with this cell embedding. And the pre-training task is very easy. They just want to predict whether those genes are expressed in this cell. So it is kind of like a binary cross entropy. They just give yes or no. And this is their pre-training objective. It's different from other foundation models. Should I move on?
Okay, so most of us actually lost.
How one focus.
那个走体，要走体，我们可能拿出来。
It's a big issue. Because the chromosome, in human we have like 44 or 46 chromosomes.
Yes.
But in other, for example, chicken, there are more. In mouse, there are fewer. And also, you're mentioning in all species. I'm not sure if they handle, for example, bacteria. There's no chromosome at all.
So you mean the input for different species the length will be different?
I think the major issue is that the chromosomes are not comparable. So it's a chromosome 1 in chicken and chromosome 1 in human. I don't think they're comparable.
But we don't need what that chromosome is. We just group them together.
Yeah.
Group them together.
So, yeah, to make them comparable, basically.
Yeah.
Group together in human, in chicken.
During pre-training, we need to when we input this cell, we first need to identify the species of this cell. we need to tell the model. And then we should find the chromosome information from the file, and we group them together.
If you can get this issue without chromosome number or count really concerned, then maybe you can consider the one and maybe more than one.
Yeah, I think there is still one issue I need to figure out is that for different species, the input length may be different. For example, maybe for oh no, sorry, the input length is the same. Sorry, they just sample 1024 genes and they group them by chromosome.
If we go back to the three problems they raised, okay, say the gene length, they said they want to gene length issue. How I can understand how their method can solve this issue.
Yeah, they don't use all genes. Instead, they just use a sample method to make the highly expressed genes to make their influence more, to give them a larger weight. And for those lower expressed genes, during sampling, the probability that they are sampled are also very, very low.
Okay, then let's say we have a gene, which is very specific, maybe only expressed in the embryonic cells, for example. But if it does not, but if in the training set there's no embryonic stem cells, then I would expect that this gene will not be sampled, right? Then
yeah, they also mentioned that after the whole pre-training, this model can be also generalized to those unseen data, and they provide some figures to prove it. But those cells, maybe they don't see it during pre-training. Yeah.
Some magic. Show us maybe. Maybe go ahead and show us.
Yeah, maybe we can first go ahead.
Regarding to the process, so finally you get one value for one cell. Since your objective was the
this is the pre-training part. Pre-training means we don't use labels of the cell. We just make it to learn some information. And actually, the output of this model, we can see that it is an embedding.
I know it is a embedding, but you mentioned that your objective is for the protein of the expressed in the cell C, right?
It's gene. It's not protein.
It's gene.
Yeah.
It's one gene.
No, it's lots of genes. It's GP represent a lot of genes.
Okay.
It's not this 20%. It's 0. GP represents the 0 expressed genes.
So you can ask the questions that are not expressed information.
Okay, so sorry, let me see. Okay, sorry. Here, they mask 20% of the expressed genes. So sorry, the sample here is from all of these genes.
All genes mask 20% of the expressed genes, right?
All of genes, sample from all of genes and plus this 20%. I think. Oh, no. Sorry.
So
The 20% of expressed genes together with some 0 expressed genes. And The objective. I may see here.
So we control the loss.
So the objective here, what's your label for the objective?
I think the tree is the same as the transformer tree. The more details. Tree. So where the difference is in the data part. Now I have a general question.
Okay.
From the three questions, three issues they want to solve. How can you explain? Can you explain from this figure which issue, what did they do to solve this these three issues? And from this figure, the first one.
First is the gene length. They
Yeah.
use the sample method.
Sample method, genus, okay.
And another is the issue. They use this protein language model.
Space. Yeah, species.
Right. And for the fine-tuning part, they claim their model, the performance is good so that you don't need to fine-tune. This is loss. Yes, loss.
So the only curve was expressed in cell state.
Yes. I think for the loss part, I may talk with UCE later. Yeah, this is a little bit difficult. Sorry.
I would have a question.
Okay.
What's the logic why they want to predict the gene P express, whether the gene P express, what's the logic? Did they give any reason?
I think for the pre-training task, generally we may have several strategies. One is maybe we mask some expression value and we predict the expression. I think it is used by Seibert. And for SCGPT, I think they also use different pre-training strategy.
Yeah, predict the next token.
Yeah, they predict next token, and here I think they just use a new strategy. Yeah.
These errors from higher. So whether they can infer the direction in.
Yeah, they just predict whether gene P express. I think it's a very normal idea. Like unlike the normal bird like lost some genes and predict this gene based on the nearby around genes, but they just predict in this cell gene P whether express them. I think it's very normal. I don't know why they can
Perform better than.
why they propose the idea. I don't know why. What's the logic? I think for example for SCG bird or SCGT, I can understand because that's a very normal
Pre-training strategy.
training
Yes,
language
yes.
model, but why they use this very normal method? Did they mention that? Well,
They didn't mention that, but
so what's the reason? What did they mention?
maybe I can't answer this question. Yes.
So GP is a lot of genes in one gene. All that is
The GP represents here.
many GP.
Yeah, so it's not one gene. But
So the objective, we just want to predict the many GPs are experienced or not.
yes, and they combined it with the embedding.
But you said object was, it's just one, but you say you use many. So it is
GP is one gene,
GP one gene.
Yeah.
And
and they say
during the back, just the back propagation, how many?
I think the GP one time GP so if for example if you have many GP, so the objective is over the GP and this
Create many
you know
samples.
GP is just a one thing or one thing.
So one sample just related to one GP as the label.
Yeah, so for one cell, they can train many times. You can sample one, you get G1 not expressed. You can present. There are many, so you can sample. GP just 就是 GP 它一次只代表一个基因，因为你是一个细胞，但是你可以把它采样多次，每次都会不一样的 GP， 但是它的表达是不一样的，就是通过你这个采样的次数，所以他说。sample GP from the cells genes， 采样很多 GP 来自这个从这个细胞的基因里采样很多次。
一个细胞不训练很多次。
对，是，我觉得是。
我觉得自己学出来挺多的。
这个就是
对，因为它是一个细胞，一次只能你一个，或者你直接采了很多次，每次再给
So this is
你一个。
binary classification.
对，他就看它表不表达。不然，
Yeah.
不然这种。
Before I agree with you, but then I said explain first. 没有 we try correct.
What's the
Yeah,
I
said
the loss of?
It's binary classification.
Yeah, it's
label
it's
classification.
binary cross entropy, binary classification.
How many rounds? For one cell?
They didn't mention it in the paper, but they gave another kinds of training details. The model they use is a 33-layer transformer with 650 million parameters, and they pre-trained on 36 million cells. And for this cell, it has around 34 million cells that are downloaded by the cell by gene corpus. It contains only human and mouse cells. And another 2.3 million cells contains this kind of 8 species cell expression values. And this is not yet published. And they use 24 180 gigabytes GPUs for 40 days. And I do a simple calculation. If they rent AWS GPU, it's around 0.3 million Hong Kong dollars. And if they use other free, maybe a less expensive online platform, is around 0.2 million Hong Kong dollars just for this 40 days for 24 100 GPU.
For the 33.9 human and mouse, what's the proportion for this?
They don't mention that, but I can check it later. Yeah, because this is we can download it. Yeah, this is from a website, and they collect maybe several hundred, 300 data sets, and they combine them together.
2.3 million 8 species. It's just the figure below.
Yeah, this 8 species.
Also include human and mouse.
Yeah, also include human and mouse. Yes, they must input the species label. And when we use it, we should also specify the species label.
Why don't they have the cat and dog species?
I think that depends on their own data set. They may only have those kind of species. Yeah. And maybe they are, I think, for example, the zebrafish, I think it's a very typical animal in biology. Yeah. And the pig, maybe is that mammal? I don't know. Yeah.
It's generated from their level.
The data, yeah, this is their secret data. But they will publish it after this paper is accepted. Yes, but since this paper is a preprint, so they won't publish the data set now. Yes. Yeah, because this also serves as another contribution of this paper, a pre-trained model and some data set.
数据集里面有4种是灵长类，一半儿。
A lot of them are mammals, maybe. Is this mammal? Is this mammal?
不
I
是
think
那下面的2个猴和上面那个鼠猴还有人都属于灵长类。
yes.
说鱼和青蛙。
Zebrafish and frog. Yeah, they are not mammals.
说的是实验，但是我不知道2个系列是不是一样。
I don't know the proportion of this data set. Yes, I think if I got the answer, I will update it. Yes, and all of the previous questions, yeah. And I think
And also you mentioned that all the species are healthy.
yeah, they don't I can't guarantee all of the cells are healthy, but
And I mean, if we have the, for example, the glioma small cell atlas, then when you use the data, you may
I think we can jump to this. We can jump to here. I also tried this on 2 data sets. One is from Dr. Zhao Zheng, and another is the mouse cell atlas. No fine tuning, just apply it. And I think maybe is this data set contains healthy and maybe not healthy cells? I'm not sure.
What's the meaning of the other different cell type?
Yeah, here the color represents different cell type, and here the color represents different tissues. This is mouse, and this is human. What's the details of this data set? UMAP, yes. Yes, I'm very surprised by this result. So I choose this paper and I want to present it to you. Yeah, I think the result is very satisfying. Especially, I also use this data set. I also use other models like SCBERT or GeneFormer, and I think the result is not very good. I think I have the details, but I forgot to add it here. I may. Yes. I think. Okay.
During the beginning part, you extract the gene from the gene is here. How do you use this for one cell? Maybe it have several. You can extract several samples. Which one do you decide as the final?
Extract several samples, several genes, right?
Yeah, one cell maybe to get several cells, several samples to generate.
One cells get several.
Yeah,
So when you do the inference,
Yeah.
Ah, yes, I think, yeah, that means that if I redo this again, the result will be different, right?
How one cell maybe use your sampling method, you will get several samples. Which one is the final one?
This is my sample methods.
Because during the training process,
Yeah, we've got a weighted sample, and if we
one sample can generate several samples during the training. Because of the sampling method. Here, you just maybe, I don't know.
I think the sampling maybe increase the robustness of the method and during inference. The weighted sampling do not deviate from each other very much.
I
You
think,
just got one time the cell embedding.
yes, no, these are different. This is for this data set. I use laser. For this data set, it's not very large. It's actually, it's a very weird phenomenon. This is around 150K, 150K, 10万 cells. And this is 1 million cells. Yes, this is 1 million cells. So if we use UCE on this data set, we just need to run it once. But for this, since it requires a lot of memory, we need to cut it into batches.
Do not use the cell embedding, just use the UMAP to do the translate.
I'm not sure whether I've got the original embedding. I'll see. No, I don't have the original embedding of this cell. It's not very fast. I think this is also my conclusion. It's slow speed. Yeah.
The inference is very slow.
The inference time is slow. For this data set, for 150,000 cells, maybe 4 to 5 hours.
Sample several times and then do the average to get the final data. So it's very slow because for the inference part, for many structure, many models, it's very quick. Because you just do the matrix computation is very fast.
Yes, actually, but for foundation models, even SC-BERT, like SC-BERT, the inference time is also slow because of the sparsity of the single cell matrix. So other than other tasks, we can pack them as the dataset format, data loader format. But for the single cell data, we can only do them cell by cell, one by one. Yeah, because the data format is different. It's a sparse matrix, and we can't convert them to data loader.
Based on the better performance, you ask the Dr. Zhao for more
Information.
information for the sample. Since they already have some like level 2, level 3, and
So we can see the more classes, right? Yeah, because they only have 4 classes. Yeah, it's okay. It's okay. I think because we've already got the embedding and we just need to assign it different labels. Okay. I will ask Dr. Zhao for more information. Okay, and yeah, I think we should still go back, and I may be quick. Yeah.
existing method to like summarize your clustering of this project.
Okay.
or gene informer.
Gene Informer, yeah, we also have the SC-BERT and Gene Informer result.
Yeah, I think that this is that's
Yeah.
previous.
Yeah, it's better than previous two model. Yes, and it's easy to use. We don't need to fine tune and we don't need to dive into the code. We just Python and give the species and give the file. It's okay.
Maybe one fundamental question or best question, why this is good?
I think for current foundation model, we can only evaluate them by the performance. Yeah, we can't know why it's
It's kind of so.
I think, but actually, this model is good. I think here is a very important reason because it utilized the large language model, large protein language model. This is designed by Facebook, and they use a lot of many protein sequences and protein to train protein embedding. I think it's kind of like another AlphaFold 2.
Why they do not just use the RNA sequence to see whether the RNA.
You mean why they use this part instead of without using this part?
Why they just use the RNA sequence AGCT instead of the amino acid protein sequence
Instead of what?
protein sequence. Your input is based on RNA sequence.
Here?
Your input, your ranking of the frequency based on the RNA sequence, whether it's expressed or not, right?
Yeah, yes.
Why you just input the RNA sequence to say the RNA is expressed in the cell or not.
Because there is no RNA expression model. They only have the ESM to do the protein.
I got your point. Sorry. I don't understand you at first. I get your point. Yes, because this part they just want to add some gene information because this part is the expression part, right? And this part is the gene part. They want to add some gene information into this model. So they have several choices. For example, SC-BERT, they use the gene to vector. That is like just have the gene interaction. And for Gene Informer, they don't use anything. Or maybe they don't use anything. I forgot. But here they use the protein language model. They just borrow some pre-trained information here. Yeah, I think that this is maybe it is a reason that their performance is best.
Maybe they can train the variables that keeps one cell. Their sample one cell many times. So
Yes, so they say that they train it for 40 days. So 40 days is 1,000 hours more than 1,000 no 960 hours. Yeah, it's very long. And during inference, it's also the speed is slow. But I think it's not that slow. Several hours for maybe. Yeah, you can also use multi GPU, but the one GPU, yes, single 3090. Yes. And I'll quickly introduce the result. The first is that after pre-training on these 36 million cells, they apply this model on the same data set. And they can get the embedding, right? And they assign the labels to the embedding, and they claim that since this data set are collected with different biological conditions, such as cell types, tissues, and others, and it also collect from different experiment conditions. So it if this picture, they just want to see that is that okay to deal with the batch effect? And can the embedding be clustered within these cell types or tissues or species like that? So they perform 4 figures to see whether they have batch effect or whether they cluster by those features. Yes. And I think this is human. I think this is human. Yes, yes. Yeah, I think this is human. Because I think human is obviously the most. Yeah. I think this is human. And the second part is to evaluate the result of their embedding. So they also compare it with 3 zero-shot methods. Means 3 foundation models. G-Informer, SCGPT, and another called TGPT. And they also compare it with some fine-tuned methods, SC Arches and SCVI. This is also a very popular method designed by UC Berkeley. And there is another version called multi-VI. It can integrate different omics together to give you an embedding. It's also a very useful tool. And this SCVI, it will only deal with the single cell transcriptome. You input the single cell transcriptome and it will give you the embedding. But those 2 methods are, you must retrain it. You need to fine tune it. And the evaluation data set is another called Tabula Sapiens version 2. This is human data from 581,000 cells and several others. And the evaluation metric is another method published in Nature Methods 2 years ago called a single cell integration benchmark. This is that this they will also release it. Yes. This is also private data set and they will release it soon. Yeah, they have a lot. And because for the Tabula Sapiens version 1, they've already published a Nature Methods or Nature something. They've already published a paper. Yes. And the evaluation metric is Nature Methods paper. And I won't dive in the complex math part, but I just briefly introduce. It has 2 parts. The first is to this is used to evaluate the embedding. And the first is the conservation of the biological variance. For example, after embedding, are the labels clustered together? And if 2 cells are similar, for example, they are all epithelial cells, are the values similar? And another is the batch removal. After embedding, will the batch effect be removed? So they have 2 kinds of evaluation methods. And the final score is the weighted average. This biological conservation counts 60%, and this batch effect removal counts 40%. And although the method is difficult, when we use it, just 2 lines, we just need 2 lines of code and we can get the score. And here they compare UCE with 3 zero-shot methods, and the raw data means they perform log normalize on the expression value, and also compared with 2 fine-tuned methods. And we may see that since the raw data is the upper bound, I think, because it's raw data, it even surpass it by 2 percentage point. Yeah, they claim that on their data set, it has the best performance. Yes, and the score is calculated by this weighted average.
different, why they after draws data. So like sorry.
Yes, actually, a recent paper also evaluate different foundation models and they give a benchmark. And as they say, the gene former is not that bad, but when we try to use it, the result is not satisfying. I think maybe there is something wrong with our usage. I'm not sure, but because that code is too difficult and we need to specify a lot of features and it's not they don't provide very detailed tutorial about that. So I think maybe there is something wrong with our usage about gene former. Yeah, maybe I'm not sure. And they also generate the embedding on both version 1 and version 2. So this is a larger data set. And this is UCE, gene former, TGPT, SCGPT, and this is the raw data. These two are the fine-tuned methods. And the first part is that we just see this line for cell type. UCE can separate those cell types. I think compared to those foundation models, it gets a better result. Because those three are, I think especially these two are mixed together. And this, I think, although it has some isolated clusters, but there are still several parts that they are mixed together. And
It's the number of clusters in the SC are
as the arch.
smaller than the UCE, because the UCE seems to have more cluster than the.
Yeah, I think for the number of clusters, it seems that the fine-tuned methods, especially SC arches, have smaller clusters. Yeah, because this is the raw data. This is the raw data. Maybe during fine-tuning, maybe they yeah, maybe they miss some information. And since version they first create the embedding of version 1, and after adding version 2, the two versions are from different donors. So they want to see after adding new donors, will it is that can cell types be aligned correctly? They just use normalize and log. Yes, they I think they don't use any batch effect removal. Yeah, because this is not the 36 million cells. It's just the version 1 and version 2.
Maybe there will be batch that they use the raw.
Raw data.
Yeah, but they don't. Really.
Yeah, I think you are right.
Data.
You are right. I think that maybe the raw data is too perfect, right? Yeah, I think it's too perfect. Maybe I don't notice that before. Yes. Because they say that log normalization are performed on the expression value. Yes. And okay, and after getting the embedding, they also create a dendrogram. Like the high level categories are labeled after, so we just see the first column. The data set is a lung tissue data set, and they use UCE to generate an embedding and create the dendrogram. And we first see that some distinct cell types like T cells, monocytes, and the endothelial cells, they are, since this embedding, they don't use labels, but after generating the embedding, they cluster together. An emergent, they say an emergent trend to cluster together. And even some high-level categories like immune cells and epithelial cells, they are also clustered together. So I think this picture, they just want to show that after the embedding, the biological condition is conserved very well. And
we assign the cell type labels to the majority of one.
After getting the embedding, they assign those labels to all of the cells.
Because there are like 20 clusters. Yeah.
Yes.
So they assign the label of the cluster by the majority of the cluster, the cells in the cluster.
What do you mean? Actually, I think those this column, the label is fixed, maybe, right?
But they cannot guarantee that all T cells are clustered together, right?
Yes, you are right.
So they assign the majority of that cluster as the label of the cluster.
I think after getting the embedding, we first do maybe like no, for each cell we have an embedding, and we give this cell a label, a type label, like a CD8 positive alpha beta T cell, like. So we only have those kind of maybe several tens of types, and we do a dendrogram. And we see that they are clustered together in the dendrogram. And these two columns, they are labeled after getting this plot. And they find that, oh, this is maybe this is monocytes, and these several rows belongs to monocytes, and these belongs to others, and they are all immune cells. Those are added labels.
You for one cell, you get 20. Then you dish the classes, so some cells cluster together, some others cluster. How did they name it? It's on the marketing. How did you know some cells cluster together? How did you know this cluster is CD positive or beta?
They don't do this clustering. I use this is that they just first get the embedding and assign each cell a label. And we can create the dendrogram.
It's not related to the cell types.
Okay, I got your point. Maybe they choose, yeah, I think maybe they choose the majority.
It's not a perfect type of
Yeah.
parameter.
I think the result is, I think it's perfect, and they also provide the code for this plot. I think that's a very good point. They for several figures, they provide the reproduced code. Yes.
They also provide the information how did they generate the data in
No, they don't provide that. They just they just use this data and then plot the dendrogram. Yeah. But for how to generate this data, they will say, we have the data set and we use UCE to generate the embedding. Yes. And finally, is the workflow to decode the new discovered cell types. And they provide three steps to discover new cell types in first is the same tissue, and then to all tissues, all other tissues. So the first part is to identify the novel cell type from the embedding space. Here we may first do like UMAP and do some UMAP, and we may see this blue part are isolated. Maybe it is a novel cell type, and we confirm it by marker gene analysis. So we first identify it, and then we want to identify it in the data set from the same tissue, right? So the second part is they use this generated embedding to train a logistic classifier. And the target is to predict whether the cell is the new cell or not. They train this embedding and use it on other data set. And then they apply the same classifier to all other tissues. And they at that time, they want to find the cell types with same pattern. Maybe they have some similar biological characteristic. And they provide a case study. There is a recently identified called kidney neuron cells, and it is identified in last year, April last year. And this cell is a long-sought erythropoietin producing cell. And it is characterized as fibroblast-like. So this kind of cell, they use the data set provided here and use UCE to generate an embedding after performing UMAP and assigning them the labels. They can see that, oh, neuron cells are isolated. So they first identify, maybe here is a novel cell type. And then they train a logistic classifier on this embedding and apply it on the previously mentioned 36 million cells. Because for these cells, this is kidney dataset, and for this very large dataset, they also contain some kidney cells. And they, after training, they predicted on this dataset and find some predicted neuron cells. And they, after confirmation with the marker gene analysis, they select the top 13 dataset with the most predicted neuron cells. And according to the paper, there are several marker genes that can be used for the neuron cells that are maybe DCN and others. And the generated embedding shows this pattern. There are 5 genes in this dataset that are highly expressed: DCN, LPAR1, COLA1, CXCL2, and CFH. Those 5 genes are highly expressed. And for another marker gene, EPO, it is always missed in dataset and lowly expressed. This is low expressed. So they mention that for this marker gene, it is not typically differentially expressed. And for another CXCL14, this column, it shows a mixed expression pattern. In some data set is high and others is low.
Comparing the genes.
Because I think this is the step 2. This is step 2. we want to confirm. First, we want to confirm the existence of this novel cell type, right? And then we want to see, according to these marker genes, are there any features of these genes? Right, and that then is the third step. They apply this classifier to other data sets, from other tissues like heart and lung and others, and they found the tissue with highest number of predicted neuron cells are those three: gonad, heart, and lung. For heart and lung, the neuron-like cells are previously observed, but till now there are no paper that reviews the neuron cells in gonad. So they claim that maybe some people can work on this part to find these neuron cells in this gonad tissue. So this procedure can reveals the novel cell type and to give us some information about whether these novel cell types also appear in other tissues. And yeah, and I will finally give the conclusion. I think I choose this paper because it's very easy to use. I think that's the most important thing, and we don't need to fine tune it. Of course not. There is a GitHub issue that someone asked, could you please give the pre-training and the fine-tuning code? And they say that they don't want to provide it because they believe their model is powerful enough, and they don't want this model to be fine-tuned on other data set. Yes, I think it's not a good response, but they insist that. Yes. And however, this UCE don't provide any downstream task demo. Like if we want to do cell type annotation, if we want to do maybe in silico perturbation, we need to write codes by ourselves. I think that is their drawback. And during inference, the speed is very slow. I think it's acceptable, but it's slow. Yeah, and there is a very high demand of memory. If we want to perform maybe 1 million cells or larger data set, we have to cut it into batches and finally combine the batches together. Yes, and I also want to try it on the bulk level data because I think the bulk input is similar. We have several bulk data, and there are also some gene expression values. There is something wrong with the code, and I'm writing an email with the author and trying to fix the bug. And till now, I tried some methods to avoid it, just skip it. Because there is a random sample of the weight, and if we use bulk data, the probabilities will, because when we use StumPy random, there we should give a probability, and it will soon goes to maybe not a number. Yeah, so that is a very weird bug, and I just write some code to skip it if we encounter that. And since I'm still training the classification, the classifier on that embedding, so this result is not provided yet. And I think the tutorial will be available maybe after Chinese New Year. Yes, if we want to use it, and I think it's very easy to use. Yeah, and I hope this tool can help you to facilitate your scientific research. Yes, and that's all. Thank you.
Question before we pause.
Yeah, maybe follow the code part for the fine tuning. So in the transformer issues, usually the fine tuning will provide the code. Why? Oh, yeah, from Ian's question, it seems that for the fine tuning, the GitHub owner will also provide the fine tuning code.
Yeah, I think we should provide the code because yeah, for future work, if you do something like that, I think you should also provide the code because those large language models or foundation models are trained with self-supervised learning, right? And they have the ability to adapt to different downstream tasks, right? And if you, when it comes to your own question, you should fine-tune it. I think if you fine-tune it, maybe the accuracy or the result will be better. And that is what we do usually. But they don't provide that. They just give the inference code.
What will happen if the authors do not provide the fine tuning code? We write
I
down
think-
ourselves.
I think nothing will happen because if this UCE can generate a satisfactory embedding, I think then maybe we don't need to fine-tune it. Yes. But if you want to use this model and the embedding is not good, maybe you may try to write it by yourself.
Yeah, looking forward for the tutorial.
Okay.
Although I do not use it, I'm curious about, as a user, will it tell us what is the input format, and what we need for the model?
In their GitHub, they don't provide that, but I think it's a common input format. But in our tutorial, I will list all of the information there. Okay. And I think that's all. Are there any questions online? Yeah, if no, that's all. Thanks.